Alright, so now it's time to finally dive into microbiology. The only other microbiology videos I have on the channel are pretty much from last year's book. They're from first day 2018, and I don't have the sketchy micro stuff or the actual book in front of the screen, right? So now what's going to be different this time around is that I will have my own personal annotated PDF on the screen. If you're looking to buy my PDF, obviously anytime, feel free to email me. My email is on the information below, usmle.live2018@gmail.com. So yeah, I'm going to be doing this entire stream with the book on the screen, going through bacteriology right now. First things first. And as I go through bacteriology, for each bacteria that we visit, I'm going to have the sketchy micro image there so you can see the final sketchy micro image and what you're supposed to recall from it. Now, I still highly urge, obviously, that you go ahead and do sketchy micro, purchase it, because it's a whole different experience when you actually get to see them drawing it while they talk about what each drawing or each part of a drawing actually represents. So that's different from what I'll be doing right now, where I'll show you the final aspect of the whole image and just the final product, and give you the story of what each aspect of the picture actually gives you. Okay, so we can get started now. First things first, basic bacteriology. Now, you do get questions on these things, right? Oftentimes you get questions that are either going to be direct on the bacterial structures, especially on the envelope and the difference between gram-positive and gram-negative, or you'll get information, where they'll tell you stuff about a bacteria, and then based off of that information, you should be able to infer other data from it. So first thing they talk about are the appendages bacteria can have. There's either going to be flagella or a pilus or fimbria. And these things are pretty much giveaways. Like if you see flagella, they're motile, right? And pilus and fimbria, these are glycoproteins, and these are literally used mostly for just adherence, right? Mediate adherence of bacteria to the cell surface. The sex pilus forms during conjugation, and we're going to see that when we get there. A spore is only found in the gram-positive bacteria. So if you ever see a bacteria that has spore to it, you know that it's gram-positive, right? So that's what I mean by taking information and being able to infer other information from it. And a spore is always keratin-like, right? It's going to have this dipicolinic acid in it. And of course, this is another thing that if it's given in a question, you can infer that's about spores. Spores allow survival. They're there for resisting dehydration, extreme heat, and even chemicals. They can actually get through your digestive tract and actually come out just fine. The last thing are your cell envelopes, right? So this is what they show you at the bottom over here. There's only two different real divisions between the two. You're either going to be gram-positive, where you're going to have the lipoteichoic acid as well, or you're going to be gram-negative, and you have something called an outer membrane, right? Two membranes, an inner membrane and an outer membrane with the periplasmic space. So let's take a look. Capsule, first things first, right? Organized discrete polysaccharide layer, except the poly-D-glutamate that you see in Bacillus anthracis. And when we get to Bacillus anthracis, you'll see in the sketchy micro image, there's a very nice button system. One of the guys is wearing a bunch of buttons that are poly-Ds. The buttons all spell out letter Ds. And capsules just protect against phagocytosis. That's so a lot of things, when they lose their capsule, now they can be phagocytosed and gotten rid of. There's something called a slime or S layer, right? The S layer is a loose network of polysaccharides, and it mediates adherence to surfaces, especially foreign surfaces like indwelling catheters, right? So a lot of bacteria, especially, all of these guys, bacteria, parasites, viruses. For now, we're talking about bacteria. So a lot of bacteria are you have to associate specific scenarios to certain bacteria, especially when it comes to pneumonia, right? There's so many different bacteria that can cause pneumonia. You really need to identify what is the most likely causative agent in specific different populations, right? Whether it be a mother, a kid in college, or it be someone in the army, or someone who's just always drinking and they're an alcoholic, right? Different scenarios present with different most likely causes of pneumonia. The next thing you see here is an outer membrane, which we just talked about, right? It's gram-negative only. They're the only ones that have an outer membrane, right? That's kind of what defines these two guys to be different from each other. So the gram-negative guy with his outer membrane looks like this, but the other one, gram-positives, they have a really, really thick cell wall, right? So you can see both of them have a cell wall. Don't make that mistake to say that a gram-negative bacteria does not have a cell wall. They do, it's just really thin, right? Whereas here you can see it's triple the size according to this image. So that's one thing you want to definitely understand. And they go on to tell you that the outer leaflet contains endotoxin, which can be lipopolysaccharide or lipooligosaccharides, embedded proteins like porins and other outer membrane proteins, and an inner leaflet that has phospholipids, right? And you can see all of that here. So you can see this porin protein right here. There's endotoxin, which can be lipopolysaccharides, right? And then in between the outer and inner membrane, more specifically in between the cell wall, the peptidoglycan cell wall, and your inner membrane, you have your periplasmic space, which is exactly where beta-lactamase is found. So beta-lactamase inhibitors work at that site. And I have seen questions where you get a diagram giving you the gram-negative cell membrane layout, and they're pointing at different points of this image, just like you see these lines are pointing at, and they'll ask you, where do beta-lactamase inhibitors function, or where do they carry out their abilities? And it's going to be right here, between the cell wall and the inner cell membrane. They then go to tell you endotoxin, like lipid A, induces TNF and interleukin-1 activity, right? So that's going to be fever and stuff. Antigenic O-polysaccharide component. Most OMPs are antigenic. And then the porin is the thing that's allowing you to have transport across the outer membrane. That's your little porin right here. So there's no porin in a gram-positive guy. So things just come out of that one membrane and can exit, right? But you have a big cell wall, right? Periplasm is the space between the cytoplasmic membrane and the outer membrane. In gram-negative bacteria, peptidoglycan is in the middle, which is your single cell wall right there. Accumulates components exiting gram-negative cells, including hydrolytic enzymes like beta-lactamases, right? So medications targeting beta-lactamases work in the periplasm of gram-negative bacteria. The cell wall itself, peptidoglycan is a sugar backbone with peptide side chains cross-linked by transpeptidases. It's a net-like structure that gives rigid support, protects against osmotic pressure damage, and that's what you're seeing over here. Right? It's much bigger, much more active in the gram-positive bacteria compared to a gram-negative. Last thing is your cytoplasmic membrane, which you have your phospholipid bilayer sac with embedded proteins like penicillin-binding proteins and other enzymes. Lipoteichoic acid, which is let me see right here. See, is only found in gram-positive. Lipoteichoic acid. Only extend from membrane to the exterior, right? So you can see it's on the outer part of the membrane, and then they come all the way outside. They don't extend inside though, and that's literally just about the lipoteichoic acid. This one right here, the orange one. What they both share though, these other squiggly lines, what they both share are the pilus or the pilus and the flagellum. Those things are shared by both guys there, right? So cytooxidative and transport enzymes, penicillin-binding proteins, lipoteichoic acid specifically induces TNF-alpha and interleukin-1 activity as well, just like lipid A did that in gram-negatives, right? So gram-negatives had endotoxin, right? Lipid A induces TNF and interleukin-1 activity. Gram-positive guys have lipoteichoic acid, which also induces TNF-alpha and interleukin-1 activity, right? Bacteria do not have mitochondria. Their electron transfer chain happens in the cytoplasmic membrane, which is the reason why we think that mitochondria are actually two bacteria living together. Like some bacteria ingested another bacteria, and that second bacteria that was ingested was the mitochondria. And they started living together and worked out. Pleomorphic bacteria are a third different type. These guys don't have rigid cell walls. So examples include Anaplasma, Ehrlichia, Chlamydia, Rickettsia, Mycoplasma, and Ureaplasma. Now the rest of this area just goes on to tell you the various divisions of different bacteria, and based off of those divisions, you're supposed to be able to identify bacteria in the question, right? So if you see gram stain, for example, this is the first line lab test in bacteria identification. Bacteria with a thick peptidoglycan layer retain crystal violet dye. So that's only going to be the gram-positive guy, right? Because this has a thick peptidoglycan layer. Bacteria within peptidoglycan layer turn red or pink, which is the gram-negative, with the counter stain, right? With counter stain. These bugs do not gram stain well, and these are the ones down here. So there's many ways of memorizing it. I mean, these little microbes may unfortunately lack real color, but are everywhere is a very long sentence. It doesn't really tend to do the job. The only way to really memorize, the only way to really memorize these guys is to pretty much, spend time actually identifying each one on its own. Learn what they look like, learn what they do, and then when you do that, you'll realize that there's no way you can gram stain it. Like Treponema and Leptospira are both actually visualized with electron microscopy. They're extremely tiny, so you know they're not going to gram stain. Right? Mycobacteria, one of the special things about it is the fact that it doesn't gram stain. Right? The cell wall has high lipid content. Mycoplasma and ureaplasma don't even have a cell wall. And the way you can remember that is their name has plasma, as in all they have is a plasma membrane. That's it, right? That's like a little thing you can use. Legionella, Rickettsia, Chlamydia, Bartonella, Anaplasma, and Ehrlichia are all actually intracellular, right? So intracellular things are pretty fun to test people on, because if you don't see gram stain, but they're actually supposed to be gram positive, right? And then maybe the question even tells you no bugs were identified, stuff like that, is because they're intracellular. You couldn't see them. In addition, chlamydia lacks a classic peptidoglycan because it has low muramic acid, right? Low muramic acid, muramate. So muramic acid is one of the things in this cell wall, and chlamydia doesn't even have that. So the cell wall of chlamydia is actually kind of different. Next up is Giemsa stain. So Giemsa stain, you're going to see that in the question. If you see Giemsa, they're probably talking about Rickettsia, Chlamydia, trypanosomes, which there's a many. So you can see this is what Giemsa stain kind of looks like. It's got this really like, almost pretty much like a gram thing going on. It's really pink, purplish pink. Plasmodium, Borrelia, and H. pylori. So not really something that you definitely need to know, but it's good to remember the name. If they won't show it to you as much as they'll actually tell it to you. They'll tell you something became more visible after using Giemsa stain. Periodic acid Schiff is actually pretty important, right? So this thing actually stains glycogen, right? It also stains mucopolysaccharides, so basically just carbohydrates. Used to diagnose Whipple disease because it can stain Trophorema whipple very well, right? You can see in this histological image, a lot of Trophorema whipple is being displayed. And that's probably coming from the Whipple's procedure they did to show you their GI system. Carbofuscin is almost always going to be a question about mycobacteria, right? So carbofuscin or Zeal-Nielsen stain is actually a stain for acid-fast bacteria, right? These stain the mycolic acid in the cell wall of the Nocardia and Mycobacteria, and you can also stain protozoa with it, like Cryptosporidium oocyst. Auramine rhodamine stain is more often used for screening, inexpensive, and more sensitive, right, rather than the Zeal-Nielsen. So just a lot of extra information. India ink should just ring a bell for Cryptococcus, right? Cryptococcus neoformans. Mucicarmine can also be used to stain thick polysaccharide capsules red. But, you have to see what your question is telling you. I've seen both things being presented because Cryptococcus is pretty high yield. So I've seen questions where they could either mention the mucicarmine or the India ink stain, and you have to think of Cryptococcus neoformans immediately. Silver stain is used for fungi, as well as Legionella and H. pylori. Right? Legionella and H. pylori, we're going to see when we do them. Like Legionella for Legionnaire's disease is actually on a silver-colored ship in Sketchy Micro. So that tells you a lot about the stain. And same thing for H. pylori. You're going to see why you're supposed to be able to remember this. There's going to be silver stuff in H. pylori as well. The last thing is your fluorescent antibody stain. Fluorescent FTA-ABS is actually one of the fluorescent antibodies that is... There's many different kinds of fluorescent antibody stains, but the one that is pretty popular is your FTA-ABS, which is used for syphilis. Right? Used to identify many bacteria, viruses, Pneumocystis jirovecii, Giardia, and Cryptosporidium. Next page talks about some of the properties of growth media, right? So there's selective media or an differential media. The same type of media can process both or neither of these properties. So a selective media favors the growth of particular organisms while preventing growth of other organisms. And these media often get mentioned in questions as well, which is another way you're supposed to be able to differentiate what bug did what it did. So one is your Thayer-Martin agar, which actually contains antibiotics that allow the selective growth of Neisseria, but it kills off many other guys. And you have to know the specifics of Thayer-Martin agar when it's going to come up later on, so I'll talk about it there. The indicator media or the differential media yields a color change in response to the metabolism of certain organisms. So that's your McConkey agar, right, which has a pH indicator, and any lactose fermenter like E. coli will convert lactose to acidic metabolites, causing a color change on McConkey agar. Now, again, with this kind of agar, you do need to know which color change indicates what kind of bacteria is present. You need to know those colors as well. So that's all going to come up. Special culture requirements. Now, another little attribute that you can use to identify a specific infection, right? So if you have H. influenzae in a question stem, they might say that they need a chocolate agar. And this gets tested in many ways, because factors 5 and 10 are needed, right? And factor 5 is just NAD+, which is basically vitamin B3. And factor 10 is hematin. So oftentimes for H. influenzae, they'll give you questions where, when something else was introduced, now it started growing well, and that is usually an introduction of hematin. For Neisseria, Thayer-Martin agar, like we just talked about, what's so good about this agar? Well, it selectively favors growth of Neisseria by inhibiting the growth of gram-positive organisms by using vancomycin, gram-negative organisms by using trimethoprim and colistin, and fungi by using nystatin, right? So vancomycin, trimethoprim, and colistin, and nystatin are all in your Thayer-Martin agar, but the trimethoprim and colistin do not kill Neisseria. So Neisseria will be the only thing that will be able to grow on this agar. Bordet-Gengu agar. Bordet-Gengu is really easy to remember because it has Bordet in it, so you will always remember Bordetella with it. The other guy is your Regan low medium, and when you do the sketchy microimage for Bordetella pertussis, you'll remember Regan low from there. In fact, both guys are actually sort of mentioned in that sketchy microimage, and they're different. So the Bordet-Gengu agar is just a potato extract, whereas your Regan low medium is charcoal, blood, and antibiotic in it. All right, so oftentimes, a question doesn't necessarily have to name the medium. They could just tell you what's in it, right? And then you have to know what medium they're talking about. Clostridium diphtheria also is very nicely described in the sketchy microimage that you're going to need Telluride or Loeffler medium for it. And that's also going to be in the sketchy microimage for the most part. But, the only way to really memorize these guys is either one, you hardcore just keep studying and memorize it, or you see it in questions, experience it, study the individual bugs by themselves, and then know. Tuberculosis requires the Lowenstein-Jensen agar, right? You have to know these guys very well. You have to memorize these. And pneumonia requires Eton agar, which actually requires cholesterol, right? So that might be given in a question where putting it on the agar alone didn't work, and then they added cholesterol and it started working and the bug was growing. So it must have been Eton agar. And it's easy to remember cholesterol because eating basically makes you have more cholesterol. So that's a little thing you can do there. Lactose fermenting enterics. Well, McConkey agar is all there, right? We just talked about E. coli doing its thing. E. coli is a lactose fermenter, so it's going to lead to color change. And the color change that you get is pink, right? Causing colonies to turn pink, acidic. Right? Acidic is almost always associated with pink anyway when you do the pH stuff, the pH strips. E. coli can also be grown on eosine methylene blue, EMB agar. It's specifically for E. coli. You're going to have colonies with green metallic sheen. And you do need to see what these guys look like. I have pictures as we keep going. Legionella, we just talked about earlier, where you need a silver stain. Here we're talking about the charcoal yeast extract agar buffered with cysteine and iron. Cysteine and iron. And this is also going to be described in the same sketchy microimage. They'll talk about this cysteine and iron there as well. So the media contents or other information. This media, the charcoal yeast extract, they tell you here Legionella, Brucella, Francisella, and Pasteurella all require cysteine enriched culture media, right? It's not just Legionella. So all these Ella guys basically require cysteine. And it's a nice little fact to remember. It's not that hard. I do look forward to doing that. That would be like I've always wanted to kind of just go through like I can't really stream UWorld on the screen, so I share UWorld questions in between these like lectures and stuff. But it would be nice to be able to do like maybe even like a test of sorts, right? Do a block at a time where I approach questions and talk about how I'm going to approach it. It's okay if I get the answer wrong, at least we can still learn together. So I do look forward to doing that eventually. Yeah, I'll try that for sure. Last thing here was the fungi, Saburad agar. If you ever see Saburad agar, you know they're talking about fungi. And that's just something you have to memorize one more time. Now you have the distinction between aerobes and anaerobes, right? So two different types of groups again. So aerobes are all obviously oxygen dependent. They need their oxygen to make ATP, a lot like us. And these examples are just Nocardia, Pseudomonas, Mycobacterium tuberculosis, and Bordetella pertussis. Right? And understanding this characteristic about them actually tells you a lot about them. For example, with Mycobacterium tuberculosis, you're going to get to learn that they tend to grow on the top of your lungs, right? They grow on the top of your lungs because that's where you have a lot of your oxygen, right? But not much. You have a lot of ventilation, but not much perfusion. So that oxygen just stays there, and they can use it. Right? So that's a nice way to memorize stuff like that. Reactivation of M. tuberculosis after immunocompromised or TNF-alpha inhibitor use has a predilection for the apices of the lung, which is exactly what I was just talking about right now. Okay, so that's pretty nice to remember. Nocardia, Pseudomonas, and Bordetella being aerobes, you're gonna see when we do sketchy micro images, they have a very specific symbol that they use for anaerobic versus aerobic, and that'll help you memorize that pretty well. So anaerobic guys are Clostridium, Bacteroids, Fusobacterium, and Actinomyces israeli. Right, so you can already see Nocardia and Actinomyces are always kind of compared, right? Nocardia and Actinomyces, and they're different even in this aspect, right? So Actinomyces is the one that's anaerobic. They lack catalase and or superoxide dismutase and are thus susceptible to oxidative damage. Generally foul-smelling, short-chain fatty acids, right? Generally foul-smelling, are difficult to culture and produce gas in tissue, mostly CO2 and H2. Anaerobes are normal flora in the GI tract, typically pathogenic elsewhere. So, everything is okay where it's supposed to be. As soon as it's somewhere else, it's not going to work too well. Aminoglycosides are ineffective against anaerobes because these antibiotics require oxygen to enter into the bacterial cell. A nice little fact many people don't tend to remember. Okay, so aminoglycosides will never work against like Clostridia, for example, because they're anaerobic. Not going to work against Bacteroids or even Fusobacterium, and definitely not Actinomyces israeli. Right? There's a lot of pharmacology when it comes to microbes. So you not only need to know every bacteria and virus and all of that, then you need to know what they grow on, what are the characteristics they have, how they cause infection, what infections and problems they do cause, and on top of that, you need to know the treatment. So it takes a long time to get good at micro, and you really have to do a lot of practice questions. Things like sketchy micro are a blessing for microbiology, but just a lot of reading, a lot of discussion. You can't memorize all of that just because you did one question or one reading of, first aid. So up here I wrote intracellular bacteria are protected against circulating immune factors such as antibodies and complement, but must develop strategies to survive the hostile environment inside macrophages. This can be accomplished by blocking the fusion of phagosomes and lysosomes, something that Salmonella and M. tuberculosis can do, inhibiting the phagolysosome acidification or escaping from the phagosome into the cytosol, such as something Listeria and Shigella do. They are very motile. Cytokines like interferon gamma, TNF-beta, and interleukin-12 induce a cell-mediated immune response to activate macrophage and get rid of intracellular bugs. So down here is one other distinction of anaerobes, which is facultative anaerobes. So facultative basically means they may use oxygen as a terminal electron acceptor to generate ATP, but can also just use fermentation and other oxygen-independent pathways. And these guys, the facultative anaerobes, are your streptococci, staphylococci, and enteric, right, the gut gram-negative bacteria. The other distinction we just read about right now, this information up here, was about intracellular bacteria, right? So you can either be an obligate intracellular, which means you can only survive when you're inside the cell, or a facultative intracellular, which means you can go in and out of a cell, and you'll be fine in both places. The obligate guys are Rickettsia, Chlamydia, and Coxiella. they need a host for their ATP. Facultative guys is all of these, and these guys don't need to be inside the cell, so you will see them outside the cell as well. Encapsulated bacteria are pretty unique and very, very important, especially when it comes to like asplenic patients, such as those who have autosplenectomy in sickle cell disease, right? So examples are Pseudomonas aeruginosa, Streptococcus pneumoniae, H. influenzae type B, Neisseria meningitis, E. coli, Salmonella, and Klebsiella, as well as Group B strep. And the way Sketchy Micro shows you something is encapsulated is most times you're going to see like a capsule or like a covering over something in the Sketchy Micro image. So that'll be your cue to remember that it's an encapsulated bug. The capsule of polysaccharide plus the protein conjugate serves as an antigen in vaccines. Something we talked about when we did immunology, right? Adding the protein conjugate gives you better response immunologically, so you get a stronger immunity. So encapsulated bacteria are supposed to get opsonized and then cleared by the spleen. So the spleen is the guy that takes any encapsulated bacteria, swallows them up, and kills them. Asplenics, people who lost their spleen either because they needed surgery, had trauma, or they're sickle cell patients, in which case you have autosplenectomy. Asplenics have low opsonizing ability and thus increased risk for severe infections. And those people, anyone who's asplenic, needs vaccinations to protect against Neisseria meningitis, S. pneumoniae, and H. influenzae. Right, so encapsulated bacteria vaccines. You have your pneumococcal vaccine, PCV13, right? That's your pneumococcal conjugate vaccine. It's PCV, pneumococcal conjugate vaccine. And you also have your PPSV23, pneumococcal polysaccharide vaccine with no conjugated protein. So obviously without the conjugated protein, it's going to have a weaker immunogenic capability. The other two are your H. influenzae type B, which has a conjugate vaccine, and your meningococcal vaccine, which is also a conjugate vaccine. They are conjugated with the protein. So of all of them, the only one that doesn't have a conjugated protein is your PPSV23. Some vaccines containing polysaccharide capsule antigens are conjugated to a carrier protein, enhancing immunogenicity by promoting T cell activation and subsequent class switching. A polysaccharide antigen alone cannot be presented to T cells. Right? So that's a weaker immunogenic response. The last thing on this page are your urease positive organisms, right? Urease positive. So if you're urease positive, you're able to hydrolyze urea to release ammonia and CO2. And if you release ammonia and CO2, you're going to increase pH. So it becomes much more basic, more alkaline, right? Predisposes to struvite, ammonium magnesium phosphate stones, particularly Proteus. If you ever see an ammonium magnesium phosphate stone, it's usually because of Proteus. And you have to remember what all those stones look like. And in renal, they tell you. that the ammonium magnesium phosphate stone has this like coffin lid shape, right? So you have to remember that. If a question describes to you a coffin lid shape, you know that you're talking about most likely Proteus mirabilis. Your catalase positive organisms were significant when we talked earlier in immunology. They were significant for people who had an issue with your NADPH oxidase, right? So they have chronic granulomatous disease. Catalase degrades H2O2 into H2O and bubbles of oxygen before it can be converted to microbial products by the enzyme myeloperoxidase. Right? Myeloperoxidase would take H2O2 and make weapons out of it to actually kill the bacteria. But if your bacteria has catalase in it itself, it's going to destroy its own H2O2, so your myeloperoxidase can't do its job. People with chronic granulomatous disease, who basically have a deficiency of NADPH oxidase, have recurrent infections with the following bugs that are all catalase positive. That includes Nocardia, Pseudomonas, Listeria, Aspergillus, Candida, E. coli, Staphylococci, Serratia, B. cepacia, and H. pylori. So that's really bad, right? You're already very susceptible now to a lot of bugs that you're supposed to be alright against. You're not supposed to be getting these infections. Pigment-producing bacteria are also pretty special because you can get that in the description. Right? So everyone remembers S. aureus has yellow pigment that it makes. Actinomyces israeli makes these things known as yellow sulfur granules, and they're not actually full of sulfur, but they're called that. So that's another thing you have to remember. Aeruginosa, we're going to see when we do Pseudomonas, the sketchy microimage has blue-green pigment all over the place. We also talked about this blue-green pigment in immunology when we first talked about catalase positive and chronic granulomatous disease with the NADPH oxidase. So blue-green pigmenting, because of pyocyanin and pyoverdin, in your Pseudomonas aeruginosa is actually very unique and specific. The last thing is Serratia marcescens. Right? Serratia marcescens, you could think of red sriracha hot sauce. This is the thing in America. It's also a thing in many other countries, but just in case you've never heard of it, red sriracha is just a really, really spicy hot sauce. So you could remember Serratia marcescens to have red pigment from that. The other two things here, spore forming and in vivo biofilm. Now, most questions about a biofilm have usually, in my experience, been S. epidermidis and viridans, right? You have to remember, as I told you earlier, every single bug should bring a story up in your mind, right? You should be able to think of a patient who would be a patient that would get S. epidermidis infection, and it would probably be someone with a prosthetic device, like a prosthetic heart valve, okay? They tell you here catheter and prosthetic device infections. Viridans would be someone who just got a dental procedure done, right? So they're going to present with infective endocarditis after getting their dental procedure. S. mutans or S. sanguinis. Pseudomonas aeruginosa, right? They tell you here respiratory tree colonization in patients with cystic fibrosis. Also ventilator-associated pneumonia, and we're going to talk about that when we talk about pneumonia. And the last thing is contact lens-associated keratitis, right? I wear contact lenses as well. And you have to know that it's not a good idea to go in a swimming pool while wearing contact lenses. That's not a good idea at all. That's one of the things because of Pseudomonas aeruginosa. The last thing here are your non-typable or unencapsulated bacteria, and that's H. influenzae, which can cause otitis media. Right? Remember that otitis media is usually the more common thing. Pseudomonas aeruginosa causes otitis externa, and you're going to remember that because when you do the sketchy microimage of Pseudomonas aeruginosa, there's a woman there using a device to listen to someone outside on the other side of a wall. Right? A biofilm is just some kind of surface that it's like it is a film, like a film that you use to take pictures. Right, it's a very thin sheet of basically, I guess, glycosamine and glycans, and it uses that to stick onto your heart valve or wherever, and after sticking on, it can cause infection. Last thing here are your spore-forming bacteria, also very significant. There aren't that many, and they're very easy to work with. There's realistically only two that are mentioned here, Bacillus and Clostridium. Right, some gram-positive bacteria can form spores when nutrients are limited, right? And it's a protective mechanism. When there isn't enough food and water, a spore will prevent it from dying of dehydration or heating and stuff like that. Spores lack metabolic activity, and spores are highly resistant to heat and chemicals. The core contains dipicolinic acid, must autoclave to kill spores, right? So autoclaving is a procedure, and it's done to all surgical equipment. Any surgical equipment is regularly autoclaved at a hospital by steaming it at 121 degrees Celsius for 15 minutes. Okay, that'll kill off any spored bugs that are on that surgical equipment. So you see Bacillus anthracis, which causes anthrax. Bacillus cereus causes food poisoning. Clostridia all cause different kinds of issues. Right, there's 4 different Clostridia. You have to know them all inside and out and understand their mechanism of action. So when we get there, we'll see them. But all Clostridia and both Bacillus are spore forming. All right. So now we're looking at bacterial virulence factors. Now, virulence factors are also going to be described in sketchy microimages because they kind of define how specific bacteria works. So protein A is literally just a Staph aureus thing, and we're going to see that in the Staph aureus image by sketchy micro. It binds the FC region of IgG, which is something we talked about when we did immunology. And that, by binding the FC region of IgG, it prevents it from doing its job, so it prevents opsonization and phagocytosis. So it's a very protective mechanism for S. aureus, thus allowing virulence to take place. IgA protease, you can tell from the name, pretty much breaks down IgA. right? And IgA is supposed to be there to protect your gut lumen, the entire respiratory tract kind of thing. So by cleaving IgA, it allows bacteria to adhere to and colonize your mucous membranes, right? Secreted by S. pneumoniae, H. influenzae, and Neisseria. All these guys that we know affect our respiratory tract and gut lumen. Last thing here is M protein, which is usually going to be just related to group A streptococci. Right? It helps prevent phagocytosis, which is fine, but it also causes a major issue. It shares a similar epitope to human cellular proteins, and we talked about what an epitope was. Right? When we looked at the FC region of IgG in immunology, we saw that this was actually connecting to an epitope to then cause antibodies to be formed, and you attack yourself. Well, you attack the bug that has that epitope. But this M protein has an epitope very similar to human proteins. So you get this issue of molecular mimicry, and you start, attacking your own body, right? So this is the possible mechanism through which you have acute rheumatic fever, right? Autoimmune response seen in acute rheumatic fever. And it's a very common thing in rheumatic fever. We're going to see that when we get there. Last thing on this page are your type 3 secretion system. This is a relatively newer thing. Sketchy Micro talked about it a lot, but first aid didn't really used to have a type 3 secretion system described in first aid. So this is also known as an injectosome, right? And in the Sketchy Micro images, you're going to see whatever the main protagonist in the Sketchy Micro image is holding something that looks like a syringe, right? So it's a needle-like protein appendage facilitating direct delivery of toxins from certain gram-negative bacteria to the eukaryotic host cell, right? So literally how this gram-negative bacteria is possibly making you sick. These images are just here to show you what things are supposed to look like. So you can see McConkey's agar, right? We talked about how this agar takes things that are that are able to ferment lactose and make it pink. So you see how it's pink over here, right? This is a lactose fermenting colony, whereas on the right side, these guys do not ferment lactose, so they stayed yellow, right? Point is, they didn't turn pink though. Don't associate the color yellow. Just remember the pink color is the more important thing there. And then here you have EMB agar, which is literally specific for E. coli. So of all three of them, the one that looks different is E. coli, right? E. coli gets that metallic green sheen that they were talking about. That's your metallic green right there. The one in the middle is Enterobacter, and this one over here is Klebsiella, right? They did not make the agar green. They didn't make green colonies. So very important to remember these. Basically, the color change is going to be something that's going to be shown, or they'll show you an image like this, and they'll tell you, oh, look, it turned pink. What does that mean? And you have to remember that lactose is being fermented, so you must be using some bacteria that ferments lactose. This next page, although not high yield, it's pretty easy stuff, right? If you get a question on transformation, conjugation, or transduction, right, and then the next page they talk about transposition, any of those things, you should be able to get it right, because they're very different from each other. But it's really easy to overlap different kinds of these procedures and forget the specifics that make one unique to the other, compared to the other. So first thing we look at is transformation. A competent bacteria can bind and import short pieces of environmental naked bacterial chromosome DNA from bacterial cell lysis. So some competent bacteria, right, binds and imports short pieces of environmental naked DNA. So you can see that's happening in this image, right? You have naked DNA just floating around. Some recipient cell was then able to just pick that naked DNA up, right, degrade the uncombined DNA while combining some of the other DNA. So now you have this transformed cell. So a cell transformed pretty much all by itself. And this is very unique. Transformation is very unique because, as you can tell from all these other images, they all require someone else, right? These guys are two cells. Here you have a cell in a virus, right? And then in transposition, there's no cell. This is just very DNA kind of stuff that's happening, right? So transformation. The transfer and expression of newly transferred genes is called transformation. A feature of many bacteria, especially S. pneumoniae, H. influenzae, and Neisseria. Adding deoxyribonuclease degrades the naked DNA, preventing transformation. That's pretty much what you're going to get your question on, right? Deoxyribonuclease would just degrade all this naked DNA, so there's no transformation, And without transformation, the cell won't change. Remember, if you change their DNA, they're able to make new things now, which means they might become more resistant to some of the treatment you were using. And at the same time, if you know that a cell is competent enough to actually pick up naked DNA and transform, then you can go ahead and put the type of naked DNA you want it to take, right? Maybe you have I always use the same example. So maybe you have naked DNA that can teach a cell to make insulin. So you put all that naked DNA, each one of these guys codes for information to make insulin. This bacteria might pick a bunch of them up and actually incorporate some of it into its own cell, and now it might be making insulin. So that you can use to your own benefit, because you can now give that insulin to patients. Conjugation is a whole different thing. Conjugation uses two cells or more, and usually it's going to be a thing between two cells, and it involves something known as an F plasmid, right? So there's an F plus and an F minus here, and look at what happens. The F plus plasmid contains genes required for a sex pilus and conjugation. Bacteria without this plasmid are termed F negative. So that's what the plus or minus is about. Whichever one has the plus is the one that has the pilus. Bacteria without this plasmid are F negative. The sex pilus forms on the F plus bacterium and contacts the F minus bacterium. Right, so this sex pilus was actually made by the F plus guy, and it connected with the F minus guy. A single strand of plasmid DNA is transferred across the conjugal bridge, also known as just the mating bridge. And there's no transfer of chromosomal DNA though, right? So the chromosomal DNA is up here, the plasmid is down here, and they just sent one strand of the plasmid, not the whole plasmid, just one strand of the plasmid. And now what's going to happen is both cells become F plus. Because you see, you basically had one strand in both of them, right? And then it was able to use its own enzymes to replicate that strand and become a double strand again. Alright, so the first guy didn't change at all. The first guy looks the same as he always did. The second one now has that F plus factor. Okay? That's all conjugation is in this aspect. The other one is different and a little more confusing. Here in HFR times F negative, no one is F plus. So that's the first thing you want to remember. There's no F plus denotation per se, but you're still going to see a sex plus being formed because there is an F plus thing happening. It's just not in the notation. Right? So your HFR guy is pretty much your F plus guy. And watch what happens here. So the F plus plasmid can become incorporated into bacterial chromosomal DNA. So now we are using chromosomal DNA. Right? Before, the chromosomal DNA was not affected. The pink circle stayed the same throughout the whole time. Now the chromosomal DNA is going to be affected. So because the chromosomal DNA is affected, now instead of writing F plus, where just the plasmid was sending, just the sex pilus was just sending a plasmid over, now it's called HFR, which actually stands for high frequency recombination. Right? And recombination should be the word that reminds you your chromosomal DNA is going to change. The transfer of a leading part of plasmid and a few flanking chromosome genes. High frequency recombination may integrate some of those bacterial genes. The recipient cell remains F negative, but now may have new bacterial genes. So this is why they don't use F plus as the notation, right? Because F plus basically meant you're capable of making a sex pilus, but that's not true here. Only the first guy, simply what happened here was the first guy took the plasmid into its own chromosome, right? So you can see from here where it was F plus originally, it became an HFR cell, right? So now it's HFR, which basically meant it's got the recombination thing happening already. So its own DNA chromosome has changed. Now it makes a sex pilus, and it's able to send one strand of that to the other person, and that other person actually recombined their plasmid with this new strand. So at the end, both of them are recombined. And both of them don't look anything like they did when they first started. So two different forms of conjugation. One where things just stay the same, but you made a duplicate of the plasmid and put it in the other person. Or second, where nothing looks the same and both of them changed a lot. Transduction actually requires a virus. So transduction requires a virus, and you can see that happening in both of these images over here. Right, so let's take a look. There's generalized transduction and specialized transduction. And of course, by the name, specialized is the one that's special. Generalized is not that big of a deal, but it does cause some good effects. So generalized transduction is basically, they describe it as a packaging error. So some lytic phage infects bacterium. Right, so that's your lytic phage right there. This is your bacteria. And you can see the virus is sending all of its genetic material into the bacteria. Parts of the bacterial chromosome DNA may become packaged into the phage capsid, right? So you can see that that DNA that went into the cell was then broken apart, right? And then you go ahead and make new viruses inside, right? But look at what happened. You didn't just make viruses that are clones of the original virus. You also took your own chromosomal DNA and made viruses out of them too. So now you have a new virus like this guy who's blue, right? So that's why they call it a packaging error. All this bacteria was supposed to do, this bacteria was simply supposed to be a factory. It was going to be a factory to make duplicates of your original virus. But in making duplicates of the original virus, it actually ended up also making new viruses using its own chromosomal DNA. So release of the new phage from the lysosomal cell occurs, and that can go infect other bacteria. So now you have two kinds of viruses doing their job, rather than just one. Right? So in a question, you might get like one virus, multiple cells, and then all of a sudden now you have two different kinds of viruses. That's because generalized transduction occurred. All right? The phage infects another bacterium, transferring these genes, and that's what you're seeing over here. Right, so the difference is, though, look at what happened between the effects of these two viruses. The first virus put its DNA into a bacteria and made more viruses. The second virus, which is the wrong kind of virus, the virus that has bacterial DNA in it, actually just sent over new bacterial DNA, and now that cell just incorporated it into their own body. That's it. You're not making new viruses here. So that's a very big difference that you want to understand, right? The red virus was actually just making more copies of itself. But the blue virus is not a real virus. It's just got, the viral proteins, but it's carrying the genetic information that came from a bacteria. So that bacterial chromosome is able to attach to this bacterial chromosome and make a new bacteria, a new bacterial chromosome. So big difference between what's happening, between what the red virus is able to do, which is actually the bad virus, versus the blue virus, which is a mistake. It was never supposed to be made. Right? So this latter effect here is very similar to what happened over here, right? Conjugation. The specialized one, the specialized transduction, is an excision event. Excision event. You're cutting something out. So what does that mean? A lysogenic phage, right? Not a lytic phage. Two different kinds, right? You have your lytic phage, which actually lyses things, and then you have a lysogenic phage, which also, you'll see what it does. The lysogenic phage infects bacterium. Viral DNA incorporates into the bacterial chromosome. When the phage DNA is excised, flanking bacterial genes may be excised with it. DNA is packaged into the phage capsid and can infect another bacterium. So this viral DNA entered the cell. It then combined with your cell's chromosomes. Then you made new viruses, but each one of these new viruses is different compared to the one up here. That's because when you cut this plasmid up, when you cut this chromosomal DNA up, you put a little bit of red and a little bit of green, but the original virus was all red. Right, so now each one of these 6 viruses has a little bit of red and a little bit of green, and that's different now, right? So now when this virus goes and infects another bacteria, look at what it did. It gave a little bit of red and a little bit of green. So that's what they're saying when the phage DNA is excised, flanking bacterial genes may also be excised. Right? So you were excising this little red area, but you also cut off a little bit of extra green on both sides. Right? So this final bacteria has red viral bacteria. This final chromosome has red viral DNA as well as green DNA from the previous bacteria, and then its own blue DNA. 3 different genomes are kind of combined here. All right, so this is what's really supposed to happen in transduction. That's specialized transduction. The generalized one is by mistake. You ended up making new viruses out of the chromosomes that you yourself carried too as a bacteria. When I say you, I'm talking about bacteria. And so this bacteria was not supposed to make viruses that are blue. That blue DNA was just supposed to go to waste when the cell died, but it actually made new viruses. Okay. Last thing here is transposition, which is pretty easy to remember because the word position, you're just moving the position of things. Transposing is actually jumping around as well, so that's kind of what's happening here. A jumping process involving a transposon, which is a specialized segment of DNA, which can copy and excise itself and then insert into the same DNA molecule or an unrelated DNA, right? Critical in creating plasmids with multi-drug resistance and transfer across species lines, right? So TN1546 with Van A from Enterococcus to S. aureus is just an example, and this is actually a pretty important example that shows up a lot. You can look up what the exact thing here is, but basically this kind of transposition would allow the S. aureus to become resistant to vancomycin. Right, that's kind of what's happening here. So you could see the plasmid, you see the transposon. Remember, the transposon is able to cut itself out, so it does that, and then it integrates itself into this bacterial DNA. So it's the new stuff in the middle now. That's all it is. Integrated itself, though. There's no virus. There's no two bacteria or anything. It's literally just DNA jumping because it has the capable equipment to do so. The next thing is comparing exotoxins and endotoxins. Right, so exotoxins outside, endotoxin, you have to understand what the main difference is and who releases them. Now, exotoxins can come from either gram-positive or gram-negative bacteria, but endotoxins only come from gram-negative bacteria, the outer cell membrane of most gram-negative bacteria. Exotoxins exit, right? Exotoxins come from inside and they're secreted from the cell, so that's why exotoxins exit, right? Endotoxins are from the outside. Chemistry of an exotoxin is that it's a polypeptide, but an endotoxin, as we talked about earlier, is something like lipid A, right, from the lipopolysaccharide, a structural part of the bacteria released when lysed. Right, so this is the reason why endotoxins are the reason why you get, when the bacteria is killed, you can get even more sick because you released more toxins after the bacteria was killed. And that's the Hersheimer reaction or whatever. Location of genes, either in the plasmid or the bacteriophage. For an exotoxin, the genes are in a plasmid or a bacteriophage. And we just saw what can happen with plasmids, right? Plasmids can send their information to other things, other chromosomes. Endotoxin is just coming from the bacterial chromosome. Adverse effects of an exotoxin? A lot, right? The fatal dose is just 1 microgram. Exotoxins are extremely powerful and will make you very sick. Endotoxin doesn't have many adverse effects. The fatal dose of endotoxins is very high. You would need a lot of endotoxin to get extremely sick. So clinical effects of exotoxins, as well as the mode of action of exotoxins, really depends on which toxin you're talking about. But for endotoxins, it's pretty straightforward. The clinical effects of endotoxins like lipopolysaccharide will cause fever, shock, and DIC, right, disseminated intravascular coagulation. And most of which is actually due through the activity of TNF, interleukin-1, and interleukin-6, all of which actually induce fever. Antigenicity. Well, exotoxins induce high titer antibodies called antitoxins. And you can often look for these antitoxins in a patient to see if, they're actually actively infected. Whereas endotoxin is poorly antigenic, so you can't really do anything against it, but you are going to get sick. Exotoxins for vaccination, toxoids used as vaccines, right? You're going to see a lot of this as we go through, like even with Clostridium. Like oftentimes you have to use the toxoid as a vaccine to generate antigenicity because they have antigenicity. Whereas for endotoxins, the toxoid form won't really help because even the toxoid is poorly antigenic. There's no immune response to the toxin, which is endotoxin. Exotoxins have high immunogenic response. Heat stability is not the biggest deal here, but exotoxins are destroyed rapidly at 60 degrees Celsius, except the guys you're going to get questions on, which are staphylococcal enterotoxin and E. coli heat-stable toxin. Endotoxins are very stable, even at 100 degrees Celsius. Typical diseases that come from exotoxins are tetanus, botulism, and diphtheria. But typical things that come from endotoxins are just meningococcemia, right? Sepsis by gram-negative bacteria. And you can see what's kind of happening here. So in the exotoxin, it came from inside the cell. It was exiting the cell, and then it causes its downstream reactions based off of the receptor it reached. Endotoxin is on the membrane. It's on the outside, right? And then it can go out and reach the host cell and cause release of TNF, interleukin-1, and interleukin-6, causing fever, sepsis, stuff like that. Right? But what the exotoxin does really depends on which exotoxin you're talking about. So you don't know what the downstream cellular reaction is going to be. So then they go on to tell you about bacteria with exotoxins. Now you need to definitely remember which toxin things release. But on top of that, UWorld has a couple questions, and your step one might too, where you are supposed to be able to compare, different toxins and know which two toxins coming from two different bugs act the same way. You really need to know that. Right? You can get a question where they talk about one specific bug and the specific thing it releases and what it does, and then they'll be like, what other bug releases a toxin that does the same exact thing? For example, I could give you a question about a patient with pharyngitis and pseudomembranes, right? Tell you that it's Korean bacterium diphtheria, not even make you guess, and then ask you what other bug would release a toxin that does a very similar mechanism as Korean bacterium diphtheria, and the answer would be Pseudomonas, because they both release toxin that inactivates EF2. Alright, so you really got to know that. This kind of questioning does come up a lot. Shigella and enterohemorrhagic E. coli both release this Shiga toxin kind of thing, which inactivates the 60S ribosome, right? 60S ribosome, by removing adenine from the rRNA, ribosomal RNA. Enterotoxigenic E. coli, Bacillus anthracis, and Vibrio cholerae, these guys are more for watery diarrhea, right? Enterotoxigenic, you have your heat labile, which cyclic AMP, and heat stable, which uses cyclic GMP, and they have different effects. So labile in the air, stable on the ground is a way to remember that. That's going to come up again when we talk about E. coli much later on. And this mechanism is pretty significant. Bacillus anthracis has something known as an edema factor, right? That also works very much like your heat labile toxin by increasing cyclic AMP. Vibrio cholerae, which obviously causes a lot of watery diarrhea, also uses cyclic AMP. Right, so you can see the similarities that you're going to get tested on. Bordetella pertussis has this pertussis toxin that literally just inhibits the G subunit of adenylate cyclase. And you're going to see that in the sketchy microimage as well. And this guy just causes the whooping cough, also known as the 100-day cough in adults. The next one are things that are able to inhibit the release of neurotransmitter. And that's something that Clostridia does, very much so, heavily tested, right? There's Clostridium tetani or Clostridium botulinum, two different kinds of neurotransmitter inhibition. In tetany, right, you're releasing you're preventing the release of inhibitory GABA, which then causes spastic paralysis, right? So it's like being flexed, right? Lockjaw. Whereas Clostridium botulinum prevents the release of stimulatory acetylcholine. So now you're going to have a flaccid paralysis. So you're not flexing at all. You can't flex because there's no acetylcholine. Things that lyse cell membranes. So you have Clostridium perfringens and Streptococcus pyogenes. Right? When you get questions of strep pyogenes, you're actually supposed to look for ASO titers, right? Strep pyogenes has streptolysin O, which degrades the cell membrane. So it lyses red blood cells, contributes to beta hemolysis, and host antibodies are formed against toxin, right? So ASO, used to diagnose rheumatic fever. Do not confuse with immune complexes of post-streptococcal glomerulonephritis. Immune complexes are antibody, antigen, and complement, right? So that's different. Clostridium perfringens has phospholipase or lecithinase. This gets described very nicely when you do the Clostridium perfringens sketchy microimage. You're going to see the lecithinase in that image as a phospholipid bilayer, and the bilayer is breaking because you have degradation of phospholipids. That leads to myonecrosis or gas gangrene and hemolysis. Clostridium perfringens has a double zone of hemolysis on blood agar, and the sketchy microimage does a good job reminding you of that too. Last thing are super antigens, and super antigens are going to come up early because we're going to talk about these guys pretty soon, right? Staph aureus and Strep pyogenes are some of the first few bacteria first aid discusses, and these super antigens cause shock, right? So Staph aureus has TSST1, and Strep pyogenes has erythrogenic exotoxin A. Both of them cross-link beta regions of T cell receptors to MAC2 on the antigen-presenting cells outside of the antigen binding site, leading to an overwhelming release of interleukin 1, 2, interferon gamma, and TNF-alpha, all of which causes shock. Right, so toxic shock syndrome, fever, rash, shock. Other toxins cause scalded skin syndrome, and we're going to see all the toxins and what they do when we talk about Staph aureus. In Strep pyogenes, you just get toxic shock-like syndrome, but none of the other stuff. No exfoliative toxin, no food poisoning from Strep pyogenes. So that was all your different, exotoxins, right? Endotoxins were obvious, right? We already said when we looked up here, exotoxin really depends on which exotoxin, then you'll know what the downstream cellular reaction is. For endotoxin, which comes from the cell membrane for the most part, is not being released from inside. Endotoxin is always going to cause the same effects. TNF, interleukin 1, interleukin 6, you're going to have your fever, shock, and DIC. So that's what they're going to show you here now. For endotoxins, lipopolysaccharide found in the outer membrane of gram-negative bacteria, both cocci and rods, composed of O antigen plus a core polysaccharide plus lipid A, the toxic component. That's the toxic component. Released upon cell lysis or by living cells by blebs detaching from outer surface membrane. Alright, so exotoxin was always secreted from inside, but not endotoxin. It's just coming from the membrane. Three main effects of endotoxin are macrophage activation, complement activation, and tissue factor activation, which is the part that causes DIC. So you can see that here. Right, very nice table. They're telling you your macrophage activation leads to interleukin 1, interleukin 6, TNF-alpha, and NO release, causing fever and hypotension. Complement activation, C3A and C5A, are going to cause your histamine release and neutrophil chemotaxis. C5A is one of the guys that recruits neutrophil. And you get even more hypotension here. The last thing is tissue factor, which we learn in immunology. Tissue factor is what you need to start up the coagulation cascade and cause DIC everywhere, right? Your whole body disseminated. So you're going to have excessive coagulation in your entire body, and that's really, really bad. That can kill you, which is why you see DIC or death here. And that's basically all the basic stuff that you need to understand before you actually start looking into clinical bacteriology. So I'm going to start this in the next video. I'll be back after eating, and then I should be starting that up in like an hour, I guess. It's 3:40 right now in New York, 3:40 p.m. So I'll probably start this at like 5 p.m. Yeah. So thank you for joining. Make sure you really understand all the things we just talked about, and don't waste too much time trying to memorize them. They come up a lot of times as you go through bacteriology, and you're going to see more and more of it as we keep going. So it'll stick to you.